Welcome to the next episode of the Microbiology Tube. So today we'll be talking about the Lac Operon. So if you haven't subscribed my channel, please subscribe my channel and help my channel to grow, and as well as share the video too. So firstly, the Lac Operon consists of the regulatory gene, operator gene and the three structural gene. And in between the regulatory gene and the operator gene there is a promoter site, and in the promoter site the RNA polymerase binds. So I would like to define you about the repressors. So because in the Lac Operon we'll be talking about the repression of the Lac Operon and the derepression of the Lac Operon. So it is usually necessary to know what is the repression. So repressor is a substances which act on an operon to inhibit the enzyme synthesis. So if there is the repressor, then the repressor will inhibit the enzyme synthesis. So if you look at here, this is the Lac Operon gene. So in the Lac Operon gene, so there is the I, this represents the regulatory gene. P, this represents the promoter gene. O, this represents the operator gene, and these three are the structural gene called as Z, A, Y and the A. So if there is the translation of this structural gene, it will form the polycystonic mRNA, and after the formations of the polycystonic RNA, it will go to the translation process and form the beta galactosidase, Fermius and the acetylase. So what are the functions of the beta galactosidase, Fermius and the acetylase is that, so the lactose will be converted into the glucose and the galactose by the presence of the enzymes beta galactosidase. So similarly, the lactose will be transported into the cells by the presence of the enzymes permease. But the functions of the acetylase enzymes is still unknown. So what actually happens? So whenever there is the presence of the glucose and the lactose, the bacteria will first utilize the glucose, and after the utilizations of the glucose, the bacteria will utilize the lactose. So I have divided, or the Lac Operon can be divided into the repressors and the derepressors of the Lac Operon. So firstly, the regulatory gene is responsible for making the mRNA. So this mRNA will encode for the repressor subunits, and there is the repressor subunits, and this repressor subunits form the repressor tetramer, and this will attach to the operator site. So firstly, the regulatory gene will transcribe. This will transcribe to form the mRNA. So mRNA will translate, so it will translate to form the repressor subunits, and this repressor subunits will form the repressor tetramer, and this repressor tetramer will attach to the operator gene. So if the repressor tetramer will attach to the operator gene, then the RNA polymerase will not attach with the promoter site. So if this is the RNA polymerase, so these RNA polymerase cannot be attached to the promoter site of the Lac Operon. So if there is not the polymerase enzymes, so what happens? So if there is not the polymerase enzymes, then these all structural proteins cannot make the enzymes. So all the structural genes cannot make the enzymes. So this was the repressor of the Lac Operon. This is the derepressions of the Lac Operon. So if you see here, the I, that stands for the regulatory gene, will synthesize the mRNA. So the regulatory gene will transcribe to form the mRNA, and this mRNA will translate to form the repressor subunits. These repressor subunits will attach to form the repressor tetramer. So I was talking about the repressor. So actually in this context, what was the repressor? The repressor was this repressor tetramer. But here what actually happens is that if there is the presence of the lactose in the medium, then after this lactose will inactivate the repressor tetramer. So I repeat, if there is the presence of the lactose in the medium, this lactose will inactivate the repressor tetramer. So if the repressor tetramers are inactivated, it cannot attach to the operator site. So repressor tetramer cannot attach to the operator site. So if the repressor tetramer does not attach to the operator site, then RNA polymerase will bind to the promoter site. Now the RNA polymerase, so this is the polymerase enzyme. So RNA polymerase enzymes is responsible for what? This is responsible for the transcription. So the transcription, this will transcribe the Z, Y and A, these are the structural genes, into the polycystonic mRNA, and this polycystonic mRNA will be translated to form the beta galactosidase, permease and the acetylase. So as I have already mentioned the functions of the beta galactosidase, permease and acetylase. So beta galactosidase is the functions of the beta galactosidase is to make the lactose into the glucose and the galactose, to convert the lactose into the glucose and the galactose. The functions of the permease enzymes is to transport the lactose into the cell, and the functions of acetylase is still unknown. But as I have already mentioned you, so whenever there is the presence of the glucose, and the lactose, what happens? At first the glucose will be utilized by bacteria, and at second the lactose will be utilized by the bacteria. But why this happens? So why this happens is that in order to utilize the lactose, what need to be switched on? The Lac Operon need to be switched on. So only if the Lac Operon are switched on, then the lactose can be utilized. But the glucose will switch off the lactose operon. How? So you know this adenyl cyclase, this is the enzymes responsible for conversion of adenosine triphosphate into the cyclic adenosine monophosphate. So what actually happens is that the glucose will inactivate this. The glucose will inactivate the adenyl cyclase, and if these are inactivated, then the ATP cannot be converted into the CAMP, and there won't be the formation of CAP. CAP represents the cyclic gene activator proteins, and the catabolite gene activator proteins, and this catabolite gene activator proteins won't be attached with the cyclic adenosine monophosphate, and there won't be the formation of CAMP and CAP complex. And if there is not the CAP and CAMP complex, then the RNA polymerase cannot bind to the promoter site. So what you have to remember is that for the attachment of RNA polymerase into the promoter site, we require the CAP and CAMP complex, and for the CAP and CAMP complex we need the adenosine cyclase. So if there is not the presence of the glucose enzymes, the adenyl cyclase won't be inactivated. So it won't be inactivated. So what happens? ATP will be converted into the CAMP, and there will be the formation of CAP and CAMP complex, and the CAP and CAMP complex are responsible for attachment of the RNA polymerase to the promoter site. So if the RNA polymerase are attached to the promoter site, this RNA polymerase will transcribe the structural gene into the polycystonic mRNA, and then after the polycystonic mRNA are translated to form the beta galactosidase, permease and the acetylase. So thanks for watching my video. So if you really like this video, please don't forget to subscribe my channel and share the